Let's dive in on blood vessels. Last lecture here on the cardiovascular system. First few slides here are going to just introduce us into blood vessels, a little bit of information about the different types, their structural makeup, and then we'll start, 5 or 6, 7 slides in, and then from there we'll just basically be working through coloring different arteries and veins of the body. Okay, thinking back to our last lecture, we kind of introduced blood vessels, more so just arteries and veins, but there are a third type, capillaries. All three types, arteries, veins, and capillaries, contribute to homeostasis by providing the structures for the flow of blood to and from our heart. And I always say this is the magic and most important part of what your cardiovascular system does, which is the exchange of nutrients and waste in our tissues, and that's what our capillaries provide. Blood vessels also play a role in adjusting the velocity, which refers to the speed of blood flow, as well as the volume of blood flow, which, if you think back to when we talked about blood here in our first lecture of this unit, we learned that about half of your blood is water. So by controlling the amount of water that we have in our blood, we control the volume. Volume just refers to the amount of blood that we have. So that's what velocity and volume refer to. First type, we talked about things like the aorta last time, the pulmonary arteries. Arteries, we learned, are efferent vessels of our cardiovascular system that take blood away from our hearts. Typically, that blood is oxygenated, except the pulmonary trunk, pulmonary arteries, which carry deoxygenated blood. Veins, we learned, are the afferent vessels that carry blood towards our heart, like the vena cavas, superior and inferior, as well as the pulmonary veins we talked about associated with the heart. Veins typically carry deoxygenated blood. The exception to that rule would be your pulmonary veins. And then in between arteries and capillaries are your microscopic thin-walled vessels that are the location of what we call capillary exchange, or just exchange, where nutrients are able to leave the blood and go to our tissues, or where we pick up nutrients like oxygen in our lungs, or water and glucose and other things that we absorb from our digestive tract. Waste leave the blood or enter the blood depending on where you're at. Like in our kidneys, we're filtering out waste from our blood so we can make it into urine. Our carbon dioxide leaves the blood, enters our lungs to be exhaled out. In our tissues, other places in our body, we pick up waste to be removed from those cells. And so it just kind of depends where we're at, what we're doing with the nutrients, the wastes. But all of that exchange takes place in our capillaries. Now, the three types of vessels are similar in a few ways. We've got a picture here of an artery, a vein, and a capillary. But capillaries, the third type here, are pretty unique in that they're thin-walled and only have a little bit of what we find in the other tubes. So that's why it says up here, the walls of arteries and veins are composed of 3 layers or tunics. Okay, whereas capillaries, only have one of those. Okay, we'll start with the tunica interna. Also, you'll see it listed as the tunica intima. You just add the letter L to the word interna. It tells you internal. That's where it's at, the innermost layer of our arteries and veins. It is in direct contact with our blood as it flows through the lumen, which is the space in the inside, so the opening of arteries, capillaries, and veins. The inner layer of the tunica interna, because there are sublayers to it, is simple squamous epithelium called endothelium. Simple squamous, remember, it tells you there's a single layer of flat plate-like endothelial cells. And surrounding it is the basement membrane. The endothelium is just like the endocardium that we talked about, the inner lining of our heart, which we learned about last week. Okay. The basement membrane is loose connective tissue that's on the outside of the endothelium, shown here in yellow, kind of the goldish color, which those two things are the only parts or only layers that make up a capillary. And these two layers, the endothelium and the basement membrane, are not thick at all. Okay, and we're talking about the wall of a capillary being microscopically thin. You know, like think of like a sheet of paper thin, might not even be that thick. Fractions upon fractions upon fractions of a millimeter, which enables diffusion to occur really easily. Diffusion being that stuff can pass through the walls either in or out. And there's a third part to the tunica interna, but we only find it in arteries. What we call this internal elastic lamina, that white layer that looks like Swiss cheese. It is a thin sheet of elastic fibers with holes like Swiss cheese, facilitating diffusion of materials. Okay. Surrounding the tunica interna or intima is the tunica media, the middle layer. Predominantly, it's made up of smooth muscle. This is typically the thickest layer of blood vessels, arteries and veins, especially we see that in arteries. Principal function of it is to mediate vascular tone, which is done through vasoconstriction, vasodilation. Vasoconstriction occurs when those muscles contract, and that kinks the hose up like a garden hose, if you think of it that way, decreasing the size of the lumen, which reduces blood flow through it. Whereas relaxing that muscle opens up the lumen, that's what vasodilation is, and therefore blood flow increases through that lumen. And now the terms vasoconstriction and vasodilation really specifically refer to arterial motion, whereas there's actually what we call venoconstriction and venodilation. But look at the amount of smooth muscle here compared to here. Okay? There's a lot less smooth muscle in veins than there is in arteries. So that's one big difference between the two. So when venoconstriction or venodilation takes place, it's not a drastic response as when we can, really constrict up an artery or really dilate it open. There's also, and we only see it in arteries, another layer of Swiss cheese looking like structure called this external elastic lamina, and separates just the tunica media from the externa. Okay. Speaking of the externa, that's just the outermost layer made up of elastic and collagen fibers. Your big arteries and veins will have small vessels within this layer called vasa vasorum, or that translates to mean vessels to the vessels. So these are kind of like the coronary arteries and coronary veins that we have in our hearts. We talked about that in our heart lecture, that your heart needs nutrients, especially the myocardium. The outer layer of your blood vessels needs nutrients because the blood inside is not going to be able to allow diffusion to get out to those outer layers. Overall function of the externa is just to help anchor vessels to surrounding tissue. So basically just hold them in place. Now I've mentioned kind of one big difference between arteries and veins, and that's this amount of smooth muscle. There's kind of some other things that I always want to point out with regards to comparing the two. One is the just thickness of the wall of an artery is a lot thicker. A lot of that comes from that amount of smooth muscle. And that affects the size of the lumen of an artery compared to a vein. Since veins are more thin-walled, their lumen is typically a lot bigger or wider, which means they can actually carry more blood than arteries. Okay? Right now, when you guys are sitting around listening to me talk, you're in a resting state, like 60-some% of your blood is found within your venous circulation. Okay? So over half is in your veins. So that's one big difference. Another difference, and this will be discussed here in a couple of slides when we talk a little bit more about veins, is that veins are the only vessels that have valves. Like the valves in our heart, they allow blood to only go in one direction, which is very important to promote the return of blood back to our heart, especially in places like our legs when we are upright and the effect of gravity is going to try to draw that blood back down. Those valves will only allow the blood to go this direction, not back, which, like I said, is very important in our legs. Keep blood from just like hanging out in our feet and pooling. As far as the three images go, I do expect you to know everything about them. So make sure you highlight everything or just put a big fat star on there because I want you to know it for sure. All right, what we're going to do here for the next 3 slides is go through arteries, then capillaries, and then veins, learn a little bit more about them and the different types of them that exist. So we'll start with arteries. All right. As I previously have stated, arteries are the efferent vessels typically carrying oxygenated blood, typically anyway, away from our heart. Structurally, the wall of an artery has 3 layers, like we see in any typical blood vessel that we just talked about. But a key feature is that they have a thick muscular to elastic tunica media. Due to their plentiful elastic fibers, arteries normally have what we call high compliance, meaning that their walls can stretch easily or expand without tearing in response to a small increase in pressure. So they have the ability to withstand the force of that blood flowing into them when our heart pumps, especially these first type, what we call the elastic arteries. This is what like your aorta is, the pulmonary trunk, the pulmonary arteries that we talked about, the largest arteries, the ones that are in closest proximity to the heart. These are greater than 3/4 of an inch, which is pretty good sized. These vessels can stretch without injury to accommodate the surge of blood forced into them when our heart contracts, but then they recoil. They are able to stretch like this because they contain more elastic tissue than muscle tissue. Aside from the aorta and the pulmonary arteries, your brachiocephalic up in your neck, kind of upper chest really, your common carotid up in your neck, subclavian in your upper chest, vertebral up in your neck, common iliacs, which are the 2 major ones that feed our legs, are all examples of elastic arteries. When our heart relaxes, the walls of our elastic arteries helps to force blood in a forward direction. They also smooth the jerky discharge of blood from our heart, help maintain blood pressure, and cause back pressure to close the pulmonary and aortic semilunar valves in our heart. Due to their elastic fibers, as they stretch, when blood is ejected from the heart, they momentarily store mechanical energy, allowing them to function as what we call a pressure reservoir. And then the elastic fibers recoil and convert that stored energy into kinetic energy and push the blood forward. From there, blood will be distributed to our muscular arteries. Probably can figure out why they're called muscular arteries. They've got a lot of muscle in them, smooth muscle specifically, about 40 layers, which constitutes about 3/4 of its wall's thickness. And there's much less elastic tissue than we saw in the previous slide. Much of the named arteries of the body, most of the named arteries that we'll look at in this lecture are this type. And these are the ones that distribute blood to specific regions of our body, so like our arms and our legs,
as well as other organs in our chest and abdomen. They can perform lots and lots of vasodilation, meaning they widen their lumen, or vasoconstriction, narrowing the lumen to help distribute the blood flow to the parts of the body that need it. They help propel blood onward during ventricular diastole, which is just a fancy word to describe, or fancy way to describe the time when our ventricles are relaxed. They contract when stimulated by our sympathetic nervous system. Other things they do is help stop bleeding, raise blood pressure, and shunt blood flow. As I mentioned, most of the arteries that we are going to look at today that have names are this type. From there, blood will flow into our smallest arteries, our arterioles, which have a tunica media much thicker in proportion to the lumen than that of our larger arteries. And this allows them to function as what we call resistance vessels, and that's actually kind of their nickname because they can perform high degrees of constriction and dilation. They regulate the flow of blood into capillary beds and are important regulators of blood pressure. The terminal end of an arteriole is called a metarteriole, which then tapers towards the junction with capillaries. Most tissues of our body receive blood from more than one artery. When two branches or more unite, supplying that same body region, we call it an anastomosis. With the letter I there, that's singular. Plural with an E there is an anastomoses. In our last lecture, we talked about the fact that our heart has these to prevent, part of the heart not receiving blood flow. And the same thing exists other places in our body, providing alternate routes for blood to reach a tissue and organ. We also see anastomoses, and we'll encounter them in this lecture with our veins and venules. We're not quite ready to talk about veins and venules because where blood goes from those arterioles is into our capillaries. So we'll talk about them now. So we're kind of just following the same route that blood flows as it leaves the heart. All right, reading through. Transfer of nutrients and other vital substances between blood and cells occurs in what we call capillary beds, which is just a network of capillaries. The term capillary means hair-like, and that's because they are super thin. They're also very, very small, microscopic. You can't just, dissect open the body and find the capillaries, which you can do for arteries and veins, but these guys are very, very small, are between and connect our arterial and venous sides. We talked about this on the first page, but they have extremely thin walls to aid in diffusion and exchange, typically only containing that single layer of endothelium with a basement membrane on the outside. Capillaries comprise most of the 50,000 miles of vessels in our body. Think about that statistic. We have over 50,000 miles of blood vessels in our body. Capillaries make up the bulk of them, even though we can't see them. They are the most plentiful. We can see them, but we'd need a microscope to do it, I should say. Diameter of a capillary's lumen is just wider than a singular red blood cell, allowing for ready access for gas exchange. We saw that in our blood lecture. We can see it several times here on this page as well. The more metabolically active a cell it is, the more capillaries it contains. Places like our brain, heart, and kidneys have a whole bunch. Metabolically inactive cells like bones, tendons, and joints have very few capillaries. In the lens and cornea, we could say that they are avascular because they have no capillaries. And we've talked about that before. Lens and cornea, remember, are in our eyes. Associated with the capillaries are precapillary sphincters. rings of smooth muscle that help regulate blood flow through the capillary. And they're found at the junction between the meta-arterial and the capillary. We'll talk about precapillary sphincters when we shunt blood flow here in just a couple slides with, well, I'll just show you a picture when I kind of talk about it. The three types of capillaries that exist in the body include the first type, continuous, second type, fenestrated, and then at the bottom are the sinusoids. Of the three, these are the most common type, that is continuous. I would highlight that phrase or sentence. This is what you find in the majority of your body. Places like our brain, lungs, skeletal, smooth muscles, gonna have lots of continuous capillaries. Called continuous capillaries because the endothelial cells that line them create an uninterrupted tube, because the cells are held together by way of tight junctions, which are those cell-to-cell contacts, cell junctions we've talked about before. Now, just because they are a continuous tube does not mean that stuff is not going to be able to pass through them. Stuff will easily be able to diffuse through, depending on what the substance is, either into the blood or out of the blood. We also have what are called intercellular clefts, which are these just very, very fine kind of cracks between the two cells where stuff can squeeze through, but not just anything. It takes something very small, like molecules of glucose. Okay. The second type are quite a bit leakier than our continuous capillaries because they have tiny little pores or fenestrations, which is where their name comes from, fenestrated capillaries, making them kind of, I always think of them like a strainer that you use when you're making, pasta after you boil it. You dump the boiling water and the pasta into the strainer. Water is going to easily pass through the holes of that strainer, just like they'd be able to do here. Whereas the bigger stuff, your noodles, which would be like your red blood cells, are going to have to stay because they're too big to fit through those holes. So they're more permeable, which just means stuff can easier or can pass through them more easily. And that is why we find them in areas where we have to transfer a lot of substances into or out of a capillary. Places like our kidneys, which is where we're filtering our blood, making urine, pulling stuff out of it, a lot of water, a lot of wastes in order to get those things out of our body. And then the villi of our small intestine, we'll be talking about that here, or those in our next unit, which is the site where we absorb most of the stuff that we ingest, which includes water and glucose and proteins and that kind of stuff. That's where we bring those things into our body. The final type, kind of another thing that looks like Swiss cheese here, capillaries that have these just huge gaps in them. We call them sinuses, which is where the term sinusoids comes from, or discontinuous capillaries, which are just irregular blood-filled spaces found only in kind of three major places: our liver, our bone marrow, and our spleen, which are places where we either bring stuff into the blood or remove it from the blood. And what we're able to do is move blood cells actually in or out. Depends on what we're doing. We've, for example, learned that our bone marrow is where we're making our blood cells. Well, that's how blood cells are able to enter the circulation. They can squeeze through these big old holes. In our spleen, we learned that's where we can remove worn out red blood cells and platelets and that kind of stuff. That's where that's going to take place. We do some of that in the liver. And our liver is where we also make our big plasma proteins that are sometimes too big to pass through other things into our blood. So these allow for big stuff to pass, whether those are actually large molecules or actually just cells. And like I said, only find them in the liver, bone marrow, and spleen. As far as the pictures, I'm not going to have you worry about identifying like the parts of those capillaries. What I definitely do want you to know is just to be able to identify them as, this is a sinusoid, this is a fenestrated capillary, this is a continuous capillary. So might put all pictures on there and ask you, which is which. So make sure you can identify them and tell them apart from one another. All right, and then finally from our capillaries, blood is going to flow back to our heart by way of veins,
beginning with the smallest veins called venules, eventually to our biggest veins like the inferior and superior vena cava, pulmonary veins, your coronary sinus that we talked about in our last lecture. There it is. I thought there was one more thing to fill in. Couldn't remember where it's at. Thank you. Okay. So as previously mentioned, veins are the afferent blood vessels that deliver blood back to our heart. Several types and features associated with veins exist. Let's first talk about venules. The smallest veins, there's actually two types of them, postcapillary and muscular. Postcapillary, if you remember, post means after. These are the venules that come directly after and receive blood from our capillaries. So they're the smallest of our venules, measuring 10 to 50 microns in diameter. Remember, microns, we're still dealing with things that are microscopic in size, very, very tiny. They're very loosely organized with their intercellular junctions, and thus they're very porous. Not pores and they don't have any money, but have holes in them essentially, which is kind of like what we just talked about with those capillaries. And actually, postcapillary venules function as significant sites of exchange of nutrients and wastes. So we do some exchange, not just in capillaries, but as well as here. And then WBC immigration refers to the movement of white blood cells, WBCs, from our blood. So this is how like white blood cells can leave the blood, something that we have to do like if we have, you know, a bacterial infection somewhere in our body outside the blood. Muscular venules are larger venules ranging in size from 50 to 200 microns in diameter. Once again, still dealing with microns, so we're still itty bitty, talking about microscopic in size. Thicker walled than our postcapillary venules. A lot of that is due to the amount of muscle they have. They do have all three layers that we find in other veins, but are still porous enough to allow white blood cells to pass through. The thin walls of both postcapillary and muscular venules allows for them to easily expand and serve as excellent reservoirs for blood accumulation. Now, we saw that there were like three different categories as far as size goes. For arteries, we learned that there's kind of three different types of capillaries. We don't really necessarily have kind of these different categories for veins other than those two venules we just talked about. Veins, you know, do increase in size from small to medium to large, but their structural changes are just not as distinct. So we don't have these kind of groupings like we do with the arteries. In general, veins have very thin walls relative to their total diameter. We talked about that on the first page, ranging in size from 0.5 millimeter in our small veins to 3 centimeters. In our largest veins, like the vena cava, is 3 centimeters. Would be over an inch because 2.54 centimeters are in an inch. So, you know, we're talking about pretty good sized blood vessels like the vena cavas. Many veins, especially those in our limbs, also contain valves, preventing gravity from pulling blood back to our feet. As you can see, we've got a picture here of two veins. We've cut them in kind of different angles. The transverse section here to show kind of the two flaps that form the valve. Here we can see it's kind of like that first page where we showed it kind of in that longitudinal section. Not something I'm going to ask you to identify as far as the pictures go, but valves are formed from folds of the tunica interna. In our previous lecture, we talked about an example of what we call a vascular sinus or venous sinus, a vein with a thin endothelial wall having no smooth muscle to alter its diameter. In a vascular sinus, the surrounding dense connective tissue replaces the tunica media and the tunica externa in providing support. The one that we talked about in that previous lecture was that coronary sinus on the backside of the heart that helps deliver blood back to our right atrium. And then there's also, well, we've actually talked about these back when we learned about the brain. what we call the durovenous sinuses that carry deoxygenated blood from our brain back down towards our heart. We have a lot more veins than we do arteries for several reasons. A lot of our veins are in pairs, accompanying medium to small size muscular arteries. These double sets of veins escort the arteries and connect with one another via venous channels called anastomotic veins. So we see, once again, I talked about it, anastomoses, not just with arteries, but also with veins. Our subcutaneous layer, which is, remember, just deep to our skin, is another source of veins. We call these our superficial veins. These are the ones that you see through your skin, like especially like if you look at the back of your hand, course through the subcutaneous layer, unoccupied, or excuse me, unaccompanied by parallel arteries. Along their course, our superficial veins form small connections or anastomoses with our deeper veins traveling between our skeletal muscles, which enables them to connect the superficial and deep veins with one another. Like I said, pictures, this page not important, nor are the pictures on this page, although I'll refer to them. Last kind of general introductory slide here before we start identification of the arteries and veins. So our arteries, capillaries, and veins are organized into circulatory routes that deliver blood throughout our body. We talked about both of these actually in our previous lecture. We learned that our systemic circulatory route, shown here in yellow, involves the left ventricle pumping oxygenated blood out into the aorta, which then eventually distributes out oxygenated blood out to our different organs. In the capillaries, we have exchange taking place. Oxygen leaves the blood, CO2 enters the blood, and that deoxygenated blood then returns back to the right atrium. Whereas shown here in blue, we show the pulmonary circulation or circulatory route, where the right ventricle pumps out that deoxygenated blood into your pulmonary trunk and then out to the pulmonary arteries, where we perform gas exchange in the lungs, picking up that oxygen, unloading the CO2, and then that oxygenated blood comes back to the left atrium. So the simplest and most common route of blood flow, and this could be the pulmonary circulatory route or the systemic, is from our heart into arteries, to capillaries where exchange takes place, then veins, and then back to the heart. But there are some sequences to this sequence, or variations to the sequence that can occur, like in the case of a portal system, which we've talked about before, and we are actually illustrating here in the middle of our figure with what we call a hepatic portal vein connecting this capillary network and this capillary network. Typically, capillaries are independent of one another, and they, the capillary networks are parallel to one another, meaning that blood flows into this capillary network, and that capillary network is not associated with anything but that artery and that vein. Whereas with a portal system, we have blood that can flow into one capillary network and then flow directly to another capillary network by either one or more portal veins. The other place that we talked about this was back up in our brain in our endocrine lecture, when we learned that there were two portal veins that connected our hypothalamus. And anybody remember what that structure was that was connected to it? Your anterior pituitary. Anybody ringing a bell on that? Okay, allowed for hormones to be carried directly from your hypothalamus to your anterior pituitary. In the case of the hepatic portal system, we'll look at that actually on a slide here towards the end of the lecture. But this enables blood to carry nutrients that we absorb from like our small intestines directly to our liver, because that's where we kind of metabolize them, process them, decide what to do with them. Over on the right is an illustration of what we call an arteriovenous anastomose, a thoroughfare channel, or a shunt. All those things mean the same thing, but what this allows for is blood to flow from an artery and skip directly across and into a vein without allowing the blood to flow out into the capillary network between the arterial and the venule. And this is because the precapillary sphincters, remember pre means before, These are the sphincters muscles that contract and close off the capillaries, preventing blood to flow into them. This is something that we do in our fingers, our toes, and our ears on a morning like today when you walk outside, especially if you're not wearing a hat or gloves, or if you're barefoot, in order to reduce heat loss and shunt that blood flow to our deeper portions of our body. So this is why your fingers, your toes, your ears are really susceptible to things like frostbite and cold damage. We don't have the blood in there to keep those tissues warm. Well, we aren't afraid to sacrifice a finger or a toe in order to protect the other more important organs. So kind of a sacrificial lamb, I guess. Like I said, pictures on this page not important for the exam. Okay, like I said, at this point, what we're going to do is for the remainder of the lecture, you can kind of scroll ahead or flip through your pages. I can scroll, you're not going to scroll. You'll see we have a whole bunch of pictures. We'll do arteries first, and what we're going to do is basically just color and identify them. In these slides, I've included some information. The bolded term is the structure of the blood vessel that you're going to have to identify, and then typically I give you some information about it, like what it's receiving blood from or what it's feeding. As far as the exam goes for these 12 or 13 slides, you're not going to have to know anything other than the pictures. So it's not like I'm going to ask you, what does your brachiocephalic feed? It's just going to be identify the brachiocephalic in this picture. Yes, sir.
What do I do if I don't have enough colors?
You just have to try to label it, I guess, or you can color them in later or whatever.
I'm about to say just wait until the lecture. Okay.
So as far as this section of the exam, the 20 questions over the blood vessels, I'd say bank on about 15 of them, because the majority of what we're doing, I mean, we spent 5 slides getting to this point, but this is, 2/3 of the lecture. So about 15 questions out of the 20 will be just, here's the picture, identify the blood vessel. Make sense? Now on the exam, I will tell you if it's an artery or if it's a vein. What I generally do, you guys know, I give you the picture, you'll know if it's an anterior view, a posterior view. I'll give you, 4 things, 4 arteries, 4 veins, whatever, 6 things. You'll have to label them. This will be another example where I give you the arrows, so you're not going to have to draw in your own arrows. So it really, hopefully, shouldn't be too bad. Now, without further ado, I'm going to flip over to my document camera and start coloring. Okay, so we're going to start with our largest arteries, the ones associated with our heart, especially the aorta, and then branches coming off of it, which we've talked about the aorta a little bit already. That's that major blood vessel, major artery receiving blood from our left ventricle. I will color it yellow because that's just what I picked up. First 2 branches coming off of it, I'm going to color are the coronary arteries. I'll color those blue. Brachiocephalic. Looks like that's kind of a reddish orange color. Dark green for the common carotids. Purple here for the subclavian. About peach for the thoracic aorta. Let's do baby blue for the abdominal aorta. Orange for your renal arteries, and pink for your common iliac. Okay, and if I get going too fast or you get lost, don't hesitate to slow me down. Now, keep in mind, before I even start coloring, I'm going to point out the fact that you know, we've removed a good majority of the organs in the chest and abdominal area. You know, the heart would be located right here, kind of where my fingernail is. The two lungs are gone. Down here below the diaphragm, the liver's gone, the stomach's gone, all the intestines are gone. Really all we've got left are the kidneys, which are very, very far back along that posterior abdominal wall. And the reason we've done that is because all these blood vessels are very posterior in the chest and abdomen. Okay, in our last lecture, I told you not to worry about identifying the different parts of the aorta when it was associated with the heart. Here we're going to kind of identify them independently of one another, starting with that aortic arch. You can see hopefully why we call it an aortic arch. It forms kind of a U shape or an arch shape. Comes up from that left ventricle and then curves right back down. Coming off of it, the very first two branches are the coronary arteries that feed the left and right sides of the heart, something that we talked a little bit about in our last lecture. Now, since your aorta curves around and comes down more on the left side of the chest, there's a singular blood vessel called your brachiocephalic, letter C. That connects to letters E and D. Whereas over here, you can see E and D are connected directly to the aorta. Letter D, fed by that brachiocephalic on the right side and fed by the aortic arch on the left side, is your common carotid. Common carotids, I should say. There's two of them. These are the ones you like take your pulse in your neck. The term common, we'll see a couple different times. Like the common iliac at the bottom means that there's going to be a fork coming off of it. We'll see when we get to the head, we have an internal and external carotid artery. Letter E, fed also by that brachiocephalic, is your subclavian. This is a major blood vessel that eventually feeds your arms. On the left side of the body, it comes directly off of the aorta. All right, back to the aorta. Now that we've created the arch by creating the U-turn or performing the U-turn, we'll kind of change and call it the thoracic aorta as it travels down through our thoracic cavity. Kind of lose sight of it. It actually passes through the diaphragm. We kind of lose sight of it until right down here where it's now going to be called the abdominal aorta. The abdominal aorta, because it's in your abdomen. Now, in a couple slides, we'll look at like letter L and, letter T that feed our abdominal organs, like the, stomach and intestines and that kind of stuff. Since those aren't included, those organs, the only branches there that I'm going to have you color and identify are these we call the renal arteries, which might seem not super important. Kidneys are real small organs compared to other organs in our body, but a very large supply of your blood, especially at rest, goes to your kidneys, because once again, that's where we filter our blood. And then finally, the very end of our abdominal aorta, it forks to form your common iliacs. These are the major leg feeding blood vessels. Like I said, common tells you there's going to be a fork in the road, and there actually is the forks. External, internal iliac arteries. We'll talk about those and identify them
in a bit.
Okay, any questions? All right, let's move on to our next set of blood vessels. Blood vessels that feed the heart, and we're discussed a little bit after in our heart lecture, what we call coronary arteries. Color of the right coronary artery, letter A, yellow. Well, pink for the muscular branch coming off of it. We'll go baby blue for the right marginal branch. About peach for the posterior interventricular branch. Left coronary artery, I'll go purple. And that pea green there for the anterior interventricular branch. And let's go with, no, that's too much. About this nice dark red for the circumflex. Okay, now looking at our picture, we've only got one image here. This is an anterior view as it states, but there are some of these blood vessels that are more posterior. See these portions of our picture that kind of look like a zipper? That represents the location of that blood vessel on the posterior side. So where it would be at on the back side, I don't have a posterior view for you to have to worry about. So your right coronary artery is one of those that is obviously on the anterior side as I colored it, but then it curves around like so to the back side. Coming off of it is a set of blood vessels. These we call the muscular branch. Run inferior to the right coronary artery. There's also a right marginal branch that feeds the kind of anterior side of the right ventricle. And then on the back side, running down that posterior interventricular sulcus is a posterior interventricular branch. Okay, on the left side, coming off the aorta, we can only see it because we've cut away the pulmonary trunk. Zip deep to that is your left coronary artery. Notice it's a really short artery. Really big one though, as far as width. It forks and forms this big one here. Anterior interventricular branch that runs down your anterior interventricular sulcus. This is one that feeds a good portion of our left ventricle. See the circle here? They're showing what can happen. You get that blocked off of myocardial infarction, heart attack. My guess is you get a heart attack in that, they call that the widowmaker for a good reason. Very, very deadly. Other branch off of the left coronary artery is this one that circles and flexes around, hence the name. your circumflex branch. Okay, any questions here? All right, in our head and neck, since this is a right side, we have a brachiocephalic. If this was the left side of the head, we would not have a brachiocephalic. I'll color that this kind of teal color. Two branches coming off of it are the subclavian and the right common carotid. Subclavian feeds our vertebral artery. Right common carotid, as I told you, common tells you it's going to fork. Forks into an internal carotid that is a big feeder of our brain. And a external carotid that is more superficial and feeds our scalp and outside of our body. Coming off of it, the facial artery that feeds our face, hence the name. Pretty straightforward there. An occipital artery goes to the back of our scalp, also known as our occipital region, hence the name. And then your temporal artery runs up the side of our scalp, past your temporal bone. Okay, as I said, we would only find the brachiocephalic on the right side because the common carotid and the subclavian on the left side come off of the aorta. Subclavian, letter B. You've already colored once, this big blood vessel there. Not only does it feed our arms, but it also feeds our brain because of the vertebral artery that comes off of it. Now here, the dotted lines that look like zippers represent the deep vessels. So the vertebral artery becomes one of those zippers because it runs internally and feeds kind of the back of our brain. If you remember a little while back in the semester when we learned about cervical vertebrae, we learned that they had extra holes in them. Those extra holes, those transverse foramina, are where that vertebral artery runs up in our neck. We also see vertebral veins coming down. We'll get to those later in the lecture. Okay. Now, letter E, letter I, those other blood vessels there coming off of your subclavian, I'm not going to worry about you knowing. So I'm not making this too terribly awful, I hope. Common carotid that we've already colored once runs up our neck, and then it comes to a fork. One of those is the external, one of them is the internal carotid artery. Letter M that I'm coloring now is the internal, runs up, very important feeder of our brain. The external stays superficial. And there are many branches coming off of it, as you can see. The only ones I want you to worry about are the facial artery, letter R. This one runs through our cheek, up around our chin, and up there by our eye socket. You get lucky, sometimes you can feel your pulse in that one. Right up in this area. Hopefully you can see why we call it a facial artery. Where's your occipital region at? That's your optic. Occipital is the back of the skull, where that occipital bone is, as well as this occipital artery. Once again, proving my point that at the beginning of the semester, I told you, learn these things now. They will help you the entirety of the semester. And then this big one here, which actually has two branches, not that you need to worry about them. Are the posterior temporal artery. And you can also feel your pulse in that one once in a while, if you know where to feel for it. Any questions on this one? All right, moving on down the line. All right, I want to talk about some blood vessels that feed our brain. We've got a picture of the brain. This is an inverted brain, so the brain's been taken out. It's flipped up upside down, sitting on a table. Here we can see like the cerebellum. Here's your brain stem. Here will be the frontal lobes. Okay. So 2 sets of blood vessels that feed our brain include your internal carotids that we just talked about, which run up from the common carotid. Coming off your internal carotid is an anterior cerebral. A couple of them actually. There's 2 of them. And then 2 middle cerebrals. The other set of vessels that feed more of the backside and inferior part of our brain is our vertebral arteries. Color those pink, and those feed orange, a basilar artery, and that feeds our posterior cerebral arteries. Okay, now as far as the common carotid and the vertebral artery here, or excuse me, the internal carotid and the vertebral, looking at our picture, letter A on both sides of that optic chiasm is where it's at. It's really misleading, showing you there's not much to it, right? Well, remember your internal carotid runs up through your neck like this, so that's why I kind of include this other picture. Just remember, all you're seeing here is like that much of it. It's been just cut. So we only see like the very ends of those internal carotids. We see kind of the same thing for the vertebrals. Hopefully pretty common sense where your anterior cerebrals are going to be and where your middle cerebrals are. Anterior runs anterior, middle's in the middle. There's your anteriors in yellow, very kind of intertwined with your olfactory bulb and olfactory cranial nerve number 1. Middle cerebral here is letter D. Oh, that's unfortunate. Now, we can see it pretty well here, left side of our image, because as you can see, the temporal lobe's been kind of cut away. Over here, where the temporal lobe's intact, all we can kind of see are these very distal parts of it. So it's a little harder to find the whole thing. Because it's in those grooves between the lobes. All right. That's kind of the set of blood vessels I want to talk about for the front of the brain. For the back of the brain, coming up through our vertebrae are those vertebral arteries. Letter F, run, remember, through our cervical vertebrae, transverse foramina up from our subclavian, or in this case, subclavian. All right, now letter F here in this picture, notice on both sides, they come together, which is kind of unique. Typically, blood vessels, like back here, they stay independent of one another. So these are actually converging and dumping blood into one. This blood vessel that runs up the front side of the pons, letter G, it's called a basilar artery. And then the only one that I want you to know coming off of it is a posterior cerebral artery. We see it really nicely over here on the left side because we've cut away, as you can see, just about half of the cerebellum. Over here where the cerebellum's intact, we still see some of it, just not as much. It's in that little groove between cerebellum and the cerebrum. Any other vessel there you see, like, letter H from letter I, letter E, those aren't going to be, well, I guess letter I, sorry, I was looking at the wrong one. Letter H, not worried about. These are all just superficial. Obviously, some of these blood vessels are going to be more internal to the brain. You can't just feed the entire brain from the outside. You have to penetrate inward, and your internal carotids are really important for that. But I'm not going to get any internal business for you guys. Okay, one of our most complicated figures here is at the bottom of this page, jumping down into the abdomen, all the way down towards the end of the digestive tract, rectum and anus there at the very bottom. We have sets of blood vessels that come off of our, and I really could say abdominal aorta here, to feed all those different organs in our abdominal pelvic area. As far as the exam goes, there's just these four that I'm going to worry about: the aorta, and then the three that are bolded: celiac trunk, superior mesenteric, and inferior mesenteric. So I'm being pretty easy on you here. Figure on the left is kind of a simplistic view of what these three major branches in our abdomen feed. Now, they don't directly feed them, as you'll see over here, but they do have the ability to feed things like our spleen and stomach and stuff. So I'm going to color them both. This image that I'm coloring here first on the left, not the one that I'm going to want you to know. It's not anatomically correct. We always try to be anatomically correct. Over here on the right side, your aorta is really, since most of those organs are intact, we can't see a good portion of it. You just see little snippets of it here and there, the top and bottom. Okay, first branch coming off of it, your celiac trunk, and as you can see, coming off the celiac trunk, hepatic is a term that always refers to liver. Hepatic artery feeds your liver, and then there are branches that come off of it. Gastric's a term that always refers to stomach. And splenic obviously refers to spleen as another branch. So as you can see, these arrows are just showing you the different organs that are fed by this celiac trunk. But over here on the right, zooming in here, it's kind of misleading with all those arrows on the left, because if you look letter B, if you blink, you'll miss it. That's it. So it's a small artery in length, but it has a big reach, 3 major branches coming off of it, and a whole lot of organs that are fed by it. Like the liver, biggest organ in the body, not of the body. Your skin is still bigger. Stomach, which is big. Your spleen's pretty good size, among others. Okay, the superior mesenteric's a vessel that comes down off of your aorta and travels more to the right side of the abdomen. Feeds organs on that right side of our abdomen. Ascending colon, transverse colon, part of the small intestine, cecum. I kind of chose yellow on purpose here to show, because this one really kind of shows up and pops. Starts here. The top, we kind of lose sight of it behind the stomach. And then we pick it back up as it travels down in our abdomen towards the right side. And you can see a lot of different things coming off of it. M, N, O are the kind of major ones. Middle colic, right colic, ileocolic arteries that I'm not going to worry about you knowing. And then our inferior mesenteric is inferior to the superior. Crazy how that works, right? And then it feeds more of the left side of our abdomen. So the descending colon, the sigmoid colon, all the way down to the rectum. And it is that one there. So coming off of it is a left colic artery. That's letter R. Letter S is a sigmoid branch. So your sigmoid colon would be that portion that's labeled there. You can see where that's feeding. And then letter T, rectal. This is all your rectum. Hence the name rectal artery. So obviously it could be a lot worse for you. Could be easy on you, I guess.
Did you say that would be on the test?
This one, not this one. This one just to kind of show you what's being fed by those vessels. This one would be the one I'd want you to know.
Okay.
Now, before I forget, you have these images in your labeling packet. Do with them as you want and what's going to help you for the exam. If coloring doesn't help you, don't color them. You know, at least do something with them. At least draw an arrow in and label them so it shows me you didn't just skip over them. You're obviously free to recolor them all you want, but you don't have to. All right, in the arm, we're dealing with the right arm, so we have, again, the brachiocephalic to deal with. Otherwise, the subclavian would be coming directly off the aorta. Subclavian we've seen many times now. Are those too close? I'm going to change that color. I'm going to make it, I'll make it a dark one. Color over it. Let's do like what is that? Dark brown. I want them to stand out from one another. Belt orange for the radial. Peach
for the owner.
Similar colors. This is too dark red. Okay, letter A at the top is brachiocephalic. That's the one coming from the aorta. We've talked about coming off of it, the common carotid up into our neck, but that's not important to us now because we're more concerned with the subclavian. Now here's where it kind of gets tricky. Your subclavian in this area of our body is not any different than this vessel here, your axillary. You know, there's a line there to help figure us out what blood vessel we're dealing with. But the actual vessel itself is still the same vessel. It's just has a different name based on where it's located. And we know the axillary region is our armpit, right? So that's where that one travels. When it gets to our upper arm, we change its name. Still the same vessel. And becomes your brachial artery. So when you take your blood pressure in, typically, it then forks. And if, once again, you can remember radius and ulna, this is going to help you tremendously. Thumb side, radial side, where your radial artery runs. Whereas your pinky side, ulnar side, is where your ulnar artery runs. Both those kind of terminate at your wrist. These arrows represent the fact you can take a pulse in both of them. A lot of nurses use the radial artery for that. They feed these two loops in our hands. Letter S and U. I'm not going to worry about which one's superficial and which one's deep. You just kind of know them together. And then feeding those, or fed off of those, I should say, are your palmar digitals. Remember, your digits are your fingers, so that's where those come from. Any questions on those? All right, let's do your legs real quick. This will be our last, should be our last arteries. I've got an anterior view and a posterior view this time because some of these are anterior only or posterior only. It's a good dark blue. All right. terminal abdominal aorta. You know, it's not actually terminal abdominal aorta in the name. It's just the terminal end of the aorta. See it, letter A, here in our anterior view. We can't see in the posterior view, nor can we see in the posterior view. Letter B, the common iliac, which I don't know why they labeled on the left one, but they don't the right. I'll color it. Common, just like in our neck, here, forks. The kind of less important of the two for our purposes, letter C, the internal iliac. Right there, letter C, not that important compared to your external iliac, which happens to be the first one that we see in our posterior view. Kind of like in our arm, external iliac changes names. When it reaches our femoral region, it becomes your femoral artery. So it courses down our thigh. Just above the knee, it becomes the popliteal artery, which hopefully you all remember. Popliteal is a fancy word for the back of the knee, which is where this one is located. So we lose track of it here, pick it back up in our posterior view. Popliteal artery comes to a point where it forks. One of those forks is an anterior tibial artery. One of them is a posterior tibial artery. Guess which view you're going to find the anterior tibial artery in? Hopefully not, or you can use your common sense, not too difficult. Letter P. Running down the anterior side of your tibia is your anterior tibial artery. We do see a little bit of it here. It pokes through the point between the two bones, and that's where we pick it up right there. At the top of our foot, it feeds the dorsalis pedis artery, letter Q. And I'm not going to worry about you knowing any of the ones that feed the rest of your foot and toes. Just that last one there on top of the foot, the dorsalis pedis. It's on the dorsal side of your foot, hence the name. Pedis refers to foot. Posterior tibial is the other branch coming off of that popliteal artery, runs down the back side of the tibia, past the ankle onto the bottom of the foot. Branch coming off of it that I want you to know is your fibular artery, letter V, runs along the fibula. And then on the bottom of the foot, remember the word plantar always refers to the bottom of the foot. This kind of loop there, your plantar arteries. Same thing, I'm not going to worry about you knowing your toe arteries. Okay. What I want to do is show you real quick, since we're running low on time, I'm not going to continue coloring, but if you go on to Canvas, there's this link here called Vessels Coloring. You click on it. What I've done is I've just done all the coloring and scanned it. And then you can finish yourself out coloring. We got about half, or a little over half of the coloring done. So that's good. We got all the arteries done. All you have the veins left. Hopefully it's nothing too crazy for you to go through and color them and identify them on your own. I did not include this picture, these pictures on the veins of the arms. Looking at the picture, there's just not a picture where I can steal these coloring pages from. That's a good one. The arms are just loaded with veins, superficially and deep. So I included these pictures from your textbook, but I promise you, there's not going to be a picture on arm veins on the exam. So that's why it's not in that scanned document. And I'm going to jump to the very last slide because this isn't a slide to color, so I want to talk about it. You should be able to handle the rest, I think, on your own. And this kind of ties in stuff we talked about in our last lecture, along with this lecture, where we're dealing with circulation. Is it just those two blanks to fill in? Okay. Circulation, in this case, fetal circulation. Exists only in the fetus, contains special structures allowing the developing fetus to exchange materials with mama. Differs from postnatal or afterbirth circulation because that's when our lungs, kidneys, and GI organs begin to function. That's when we start breathing and eating, all that wonderful stuff. We have to obtain our own nutrients. We're not getting them through the placenta after we're born. While we're in the womb, fetus obtains O2 nutrients from and removes CO2 and other waste into maternal blood, and then mom is the one that exhales out the CO2 and pees out the waste. This is done between the placenta, which forms inside the mom's uterus, and is attached to the umbilicus or navel of the fetus by what we call the umbilical cord. Communication with the mother's CV system, cardiovascular system, occurs via small blood vessels emerging from the uterine wall. Umbilical cord contains blood vessels that branch into capillaries in the placenta. Waste from the fetus diffuse out of the capillaries into the spaces containing maternal blood in the placenta, and then into mom's uterine veins. Nutrients are going to travel the opposite direction. Blood passes from the fetus to the placenta via two umbilical arteries, which are branches of the internal iliac arteries located within the umbilical cord. At the placenta, fetal blood picks up O2 and nutrients and eliminates CO2 and waste. Oxygenated blood returns from the placenta via a single umbilical vein, descends to the liver, where it then divides into two branches. And so we'll start there. So here's the umbilical cord connected to this will be the baby's belly button. Here's the placenta connected into mom and her uterus. Inside are umbilical arteries and veins. In this case, this vein, notice what color it is red. And that's because the umbilical vein that's coming into baby is carrying oxygenated blood towards the baby's heart. Remember, red always denotes oxygenated, whereas blue denotes deoxygenated. Okay. In the liver, that blood vessel splits to form two blood vessels. Okay. So right here is where I was at. Divides into two branches. One branch joins the hepatic portal vein, enters the liver. The other branch, called the ductus venosus, drains into the inferior vena cava. So here's where it's joining the hepatic portal vein right there. Here's the other one joining the inferior vena cava. Notice what we see as far as color, though. Thinking back to when you were in elementary school, you learned that when you mix red and blue, you make purple. Not that the blood is actually purple, but they denote purple for what we call mixed blood. Partially oxygenated, partially deoxygenated. Mixed blood in the inferior vena cava, deoxygenated blood in the superior vena cava passing to the right atrium. Instead of passing into the right ventricle, the blood can pass through that hole that we talked about, called the foramen, the foramen ovale, which allows for the lung to be bypassed.
some blood does make it into the right ventricle and then gets pumped out into the pulmonary trunk, but then we have another lung bypass, a blood vessel that connects the pulmonary trunk to the aorta, what we call the ductus arteriosus. We talked about those in our previous lecture. Okay. This blood in the aorta then carries the blood to all tissues through our systemic circulation in the fetus. When blood reaches the common iliac arteries, part of the blood flows into the internal iliacs and then eventually the umbilical arteries that then go out into mom's blood. Okay. Now after birth, when the lungs, kidneys, and digestive organs begin functioning, there's no need to have some of those things. You know, for example, there's no more umbilical cord that gets cut off, so the belly button has no purpose other than to basically be a scar. So there's no need to feed it with blood, and there's no blood coming away from it. So those umbilical arteries become what we call medial umbilical ligaments, basically just fibrous cords that serve no purpose. The umbilical vein becomes a structure called the ligamentum teres. No blood's coming through it, so there's no purpose of having it anymore. The hole in the heart between the right and left atrium should close within a year or so. Becomes what's called the fossa ovalis. You can still see that when you look at the inner atrial septum from the right atrium. There's no more need to connect the pulmonary trunk and the aorta. That becomes what we call the ligamentum arteriosum. And then the final thing, and this is the first thing that takes place after baby's born, as placenta is expelled as after birth, which, you know, they use to run tests and stuff on. In some cultures, they gather it up and use it to make chili and stuff like that. Other animals eat their afterbirth. It's a natural thing. There's actually a lot of benefits to it, but it is not something that we did in our family. Okay. As far as the exams go, do not worry about the pictures. I do want you to especially focus on these last 6 things, talking about what happens and what changes occur to the cardiovascular system.